So good, you guys can make more mistakes. That's how it works. You guys know that. Yeah.
Okay, so the last SI is this coming Monday?
Yes.
And then the review is Wednesday?
And then the review is on Wednesday, yeah. All right. Let me pull up the PowerPoints. And yeah, if we don't finish 30B today, I will probably do like a video recording over the weekend and post it, because I do want to start the review on Monday. So, all right, let me make sure everything's going. Okay. All right, so what was the last thing we were talking about? We did that, we did that.
Normally in Anticodon, but in mitochondria.
The specialist situations, okay, so that's this one. Yeah, so the last thing we talked about on Wednesday were the exceptions to the universal rule for triplet code. The universal rule for the triplet code, by the way, is if you take a look at this table, you see any of these codons. Do I have to turn out one light? Probably. Where's this light switches? Back there? Is that all right? Can you guys see that? Or you guys all right? All right, then. If you guys can see it, then it's fine. Yeah, so triplet code. Each of these codons code for a particular amino acid, and it turns out every organism that we know about follows this table. There's these codons to amino acid linkage, meaning this was developed very early on in the evolutionary process. There are a few exceptions, which are listed on this slide, and they both involve the normally the stop codon UGA. One exception is that in mitochondria, the stop codon UGA actually codes for tryptophan, and also some organisms, some prokaryotes and eukaryotes, UGA actually codes for a special amino acid that is not one of the 20 amino acids called Cellino Cysteine. You have to know how Cellino Cysteine is made, which is nicely detailed here. Be very careful. I will stress that Cellino Cysteine, despite the name, is not made from cysteine. It is made from serine, confusingly. And basically, serine has a hydroxyl group on the end there that will get modified into the Cellino Cysteine, which you won't have to identify via pictures. Okay, next thing. All right, so the next thing I talk about is actual tRNAs. So if there's a nice picture to go with this, not really. So let me go back to the tRNA picture up here to illustrate. So this tRNA is the hybrid between amino acids and the genetic code that you get in mRNA. This tRNA is going to be holding on to an amino acid up here at the 3' end of its structure, at the tail end of its structure. And down here you have a region called the Anticodon, which we'll see in translation is very important, because this Anticodon is going to associate with the codons in mRNA in order for ribosomes to understand what amino acids it needs to put together. But how does a tRNA and its amino acid get put together? Because when a tRNA is made, it does not automatically come with an amino acid. There is some mechanism that exists to match tRNAs to their amino acids. So that is what these next couple slides are going to talk about. There's one enzyme that will take care of that called Aminoacyl-Trna Synthetase. Okay, so you do need to know that name, and the name of the enzyme gives away what it does. Aminoacyl-Trna Synthetase. This enzyme brings together an amino acid and a tRNA to form that structure of a tRNA holding onto an amino acid. So, how is it able to do this? Oh, well, before we get to the how part, there's one extra mechanism they talk about here, is that there's a secondary enzyme called deacylase, which edits mistakes that aminoacyl-Trna synthetase makes. So you can compare and contrast this to, your DNA polymerases. Remember that there was activities for that to proofread and edit mistakes. It turns out that at this level, at aminoacyl-Trna level of mechanism, it also has an editing mechanism called deacylase, whose job is to literally hydrolyze the amino acid off of the tRNA in order to bring in the correct amino acid. Don't worry about like all these numbers and stuff. I've never seen that be asked about. This looks like a really complicated slide, but honestly, the only thing you need to know, I will zoom in, is that enzyme, Aminoacyl-Trna Synthetase, there are two classes of it. Class 1 and Class 2, so the names are pretty easy to memorize. But the end result is the same. Either one, the end result is to take an amino acid and attach it to a tRNA. All right, so there are two different classes of aminoacyl-Trna synthetases, Class 1 and Class 2. The other part to note is Class 1 takes 2 steps, and Class 2 only takes 1 step, so annoyingly like that. So memorize it as a bit of a backwards thing. Class 1, the second step is called transesterification, which is a moving the ester bond from one place to another. Don't worry about that. That's more of like an organic chemistry thing rather than a biochemistry thing. So bottom line, there are two different synthetases. And you don't have to memorize this up here, but what this is trying to illustrate is of your 20 amino acids, some of them are attached to their tRNAs via a Class 1 synthetase, and some of them are attached via Class 2, but don't bother memorizing which one is which. Bottom line, either class is used to combine an amino acid to its tRNA. Let me zoom out some. Yeah, and some visualization if you want. Pictures. Oh, okay, so some interesting stuff with that. You might wonder, well, how does a synthetase know what amino acid to attach to what tRNA? And a lot of people will think, and logically it would make sense, that it must look, let me look for this picture here, it must look at the anticodon region, right? Because if the codon table tells us that certain codons are matched to certain amino acids, then wouldn't it make sense that synthetase will understand what amino acid goes onto a tRNA by just reading what bases are in the anticodon region? One of the strangest things is that is not true for every single amino acid. I'll explain using this diagram to illustrate that. It turns out that the aminoacyl-Trna synthetase, whose job is to attach phenylalanine onto its tRNA, does read the anticodon region. That's what this orange stuff is indicating, is where that particular synthetase is reading to understand that this is the correct tRNA to put on phenylalanine. But also notice it looks at two other places on the tRNA that have nothing to do with the anticodon. So it turns out, depending on which tRNA and which amino acid you're talking about, there are certain regions on the tRNA that communicate to the enzyme, that communicate to the synthetase that lets it know what amino acid should go there. To contrast, here's methionine, and eventually you'll see what the F means. But there's a synthetase whose job is to attach methionine to this tRNA, and it's only going to look at the anticodon region. Now we get to some weird ones. The synthetase whose job is to take serine and put it onto a tRNA, that synthetase does not care about the anticodon region for whatever reason. It only looks at the highlighted areas you see here. And in fact, because it doesn't care about the anticodon region, it's only looking at these indicated spots. So these spots are conserved spots on this tRNA that communicate to the enzyme to let it know that serine goes onto this tRNA. It's a little weird, and I personally don't know why that is. I think there's a lot of research being done to figure out why that is. But this is an example showing that the synthetase doesn't always need the anticodon to know which amino acid goes onto its tRNA. Here's a really extreme example. The synthetase whose job is to attach alanine to its tRNA cares only about two specific bases being particular bases. I don't actually know what it is. It might actually be written somewhere on the left here for you. Bottom line, it only looks at base number 3 and base number 70, and whatever bases are supposed to be there, if it finds them there, it knows to put alanine on that tRNA. So those are conserved in the tRNAs that are supposed to contain alanine. Yes?
Should we know these specific examples, or are you just trying to prove the point that they don't look at the anticodon?
You actually do need to know the alanine example. Let me make sure because that's probably in the text over here on the left. Yeah, you do need to know the alanine example because sometimes he asks a question. Basically what you see over here is that we've done experiments where we take a completely unrelated tRNA, maybe one that has an anticodon that isn't supposed to be for alanine when you go look at the table. We take that tRNA and we modify just base number 3 and base number 70, and we've tricked the synthetase into loading alanine onto that tRNA, even though it's not supposed to go there when you only look at the rules on the codon table. So that's all that info is listed here. So if you want to look at that, you can get more details there. So context-wise, more general type question will be something like, what are the ways a synthetase recognizes how to load an amino acid onto its tRNA? Answer would be multiple different answers. Anticodon region, certain bases in the loop, in the stem loop, and in the tail. All those words are listed in these couple slides talking about the tRNA, aminoacyl-Trna synthetase. Yeah, there's actually some bases have specific names. Base number 73 has the name discriminatory base, so that would be like an answer choice, is that sometimes it reads the discriminatory base. Let me see if there's other vocab words here that you need to know. The second genetic code. The second genetic code is the phrase describing this process with the aminoacyl-Trna synthetase. It has its own patterns it's looking for to match an amino acid to its tRNA. So obviously you don't have to know what that table looks like, but there is some, and it varies by organism, by the way. I think somewhere here it says it's not universal. That this particular enzyme reads certain spots on the tRNA to understand what amino acid goes onto it. I think that's it. Let's see. Oops. Okay. So yeah, you can look at those notes there for all the information that you could possibly need about aminoacyl-Trna synthetase. Getting to this topic is actually probably a little bit more important for the exam because I've seen more questions asked about the third base Wobble hypothesis rather than aminoacyl-Trna synthetase. And we'll do a practice question about this probably on Monday rather than today. But something called third base degeneracy. So let's hope there's a nice picture. There is. All right, so third base degeneracy. Remember the phrase degenerate code wasn't one of the vocab words we used to describe the codon table. The degenerate code, what that basically means is that one amino acid is usually coded by multiple different codons. The reason why that is there are, actually I'll ask, how many possible combinations do you have of 3 bases at a time? 64. How many of those, most of the time, how many of those actually code for an amino acid? 61, right? But there's only 20 amino acids, so there's way more combinations than there are amino acids. So we have that degenerate code where multiple codons code for a single amino acid. Physically, what that means is that at the part in the ribosome where a tRNA, which you now see is this loop here, is being matched to a codon on the mRNA in order to understand what amino acid up here is supposed to be brought to the ribosome. This match doesn't have to be perfect. Well, the first and second base have to be perfect. The first base in the codon and the second base in the codon have to be perfect. But this third base in the codon, known as the Wobble position, or is also the first base in the anticodon. So the third base in the codon or first base in the anticodon is the Wobble position. That one very often experiences what's called non-canonical base pairing, where the base pairing is not what you'd expect all the time. For example, let's see, that's a good example. So over here, let me zoom in. One example of Wobble base pairing is on the Anticodon, there's a uracil. And normally, we know that uracil is going to associate with adenosine, right? That's normally A's bind with U's, right? But an example of non-canonical base pairing at the Wobble position is that sometimes a uracil on the Anticodon will bind to a guanosine on the codon, on the third position of the codon. Another example is over here. We know normally G binds with C, but sometimes for whatever reason in the Wobble position, G can sometimes bind with U. And down here you even see this base that isn't a normal base called inosine. Inosine is like a modified, I believe it's a modified adenosine. You don't have to know what modification it is. It's just a special base that you only see in tRNA. This thing has the ability to base pair with 3 different bases, adenosine, uracil, and cytosine. So that is also an example of Wobble base pairing. You don't have to memorize this table, but do be prepared, and we will go over an example of this. I'll probably do this over the weekend. I'll record it. That you will have to use this table to figure out Wobble base pairing question on the exam. But breaking down how to read this table, all the first row lines are bases you find in anticodons. All the second row lines are bases you'll find in codons. So it's telling you what binds to what. All right. And of course, this table you will also be given on the exam when we want you to tell us what amino acid sequence do you get from a particular mRNA sequence. Okay, so let me make sure I didn't miss anything yet with the Wobble. Okay. Oh yeah, this is a pretty important fact here. Why even have Wobble base pairing? Why not just stick with canonical base pairing? The advantage of having Wobble base pairing is you don't need exactly perfect association between the codon and anticodon. In fact, if it's not as perfect, the tRNA can dissociate from the mRNA a lot faster after it's dumped off its amino acid. Remember, the tRNA's job is to bring in an amino acid, and as soon as it's done that job, it needs to go so that the next tRNA can come in, since this happens one at a time. This process is sped up when we have Wobble base pairing, non-canonical base pairing, because that third base doesn't bind as strongly between the anticodon and the codon. So it's easy for the tRNA to dissociate and for translation to continue. Again, the first 2 bases in the codon is canonical base pairing. Those do have to follow the rules, but the third one, it does not. This is another way the Wobble table is given to you, but we don't usually give you this version of the table. We give you that previous one that I showed you guys. So I'm just going to move beyond that. Let's see here. Oh, this I will go over on the example that I'll cover over the weekend. But this, because of that Wobble base pair table, you can calculate the minimum number of tRNAs that will be needed to match to all the codons. Because of the Wobble base pairing, you don't need 6 different anticodons for leucine. If you go look in that codon table, you'll see there's 6 different codons for leucine. But because of Wobble base pairing, you will not need 6 different anticodons. Because the third base is a little weird. It can bind to a bunch of non-canonical bases. So the spoiler here is that you only need 3, but I'll go over how that is determined when I go over an example over the weekend. Yes?
Is that the chart we're going to be given on the exam?
Not these 2. The one that you'll be given for Wobble base pairing is going to be this one right here. Whoops. It's going to be this one right here.
And that's okay. Per amino acid or just in general?
This is per codon. So okay. If you look at each codon, at the third base, you look at what's, if you're given the, and most likely you'll be given the mRNA. So you look at the third base in the mRNA, and if you find that it's a G, you go look everywhere on this diagram what anticodons can match to it. So starting up here, if the third base on the mRNA is a G, we know it can bind to C, that's normal. We go down here, if the third base is a G, we know it can bind to a uracil sometimes, and that's everything. So you have to look at whatever base they're talking about. Alright. Let's see, I want to get to the translation mechanism, so we can go over that now rather than later. Okay. And last is just a reiteration of ribosomes in prokaryotes and eukaryotes. You see these notations for ribosomes. They really are just notations describing the large and small ribosomal subunits, because ribosomes come in two pieces initially that have to come together to form the whole ribosome. Out of all this, the only facts that I've seen them asked about are based off these last 3 bullet points here about the ribosome. One interesting fact is that the ribosome is actually mostly RNA, not protein, and that a ribosome is a significant portion of a cell's mass. And that makes sense. The cell has lots of different proteins that are being synthesized, so you need to have a lot of ribosomes doing that. And of course, in eukaryotes, because we have all our different organelles, ribosomes are found in a bunch of different places. In the cytosol, there are separate ribosomes in mitochondria and chloroplasts, which is interesting. And of course, we have ribosomes on the endoplasmic reticulum. That's why we call it the rough endoplasmic reticulum, because it's coated with ribosomes. Okay, now we get to, I think, the cool part, which is the mechanism of translation, which is covered in 30B. Okay, so 30B, most of it you can cover by following this link. So I'm actually going to use this to illustrate translation. Did he do this one yet? Oh my god, really? That's not good. 1 second. Adobe Flash update. I wonder if it'll let me. That's not good. I wonder if FE will let me do this. I thought they all came with this, but I guess not. If not, then this will be a lot more boring. Oh, okay. Because I don't think it'll let me. Oh shit, it's downloading other stuff. I hate that. Every time Adobe tricks you into downloading a bunch of extra crap. Okay, is it done? Yeah? Alright, I'm going to close that then. Yes. Finish. Okay. I don't care about other products. Thanks. Okay. Click to enable, yes. Thank you. Alright, that wasn't too bad. Alright. A very good animation describing the different steps in translation. So we'll go through this, and you'll see that once you go through that, you can skip quite a lot of 30B. So, let us go step. Obviously, you know that in eukaryotes, translation goes on in ribosomes. Is there a way to skip to the next thing? Let me get to the cool stuff. Blah, blah, blah. Ribosomes are everywhere, or whatever. Okay, so translation is split up into 3 steps. You might recognize these names: initiation, elongation, and termination. So it's the same names as in transcription, but they each involve different components, of course. So initiation, of course, is the assembly of the ribosome and all the pieces you need to do translation. Elongation is the step-by-step repetitive process of adding amino acids to an extending chain via bringing in a bunch of tRNAs one at a time to match codons, and you'll see the animation in detail later. Termination is once you've built your protein, however long the amino acid sequence is the disassembly of everything. Disassembly of the mRNA from the tRNAs, of the amino acid sequence from everything, of the ribosome subunits from each other. Everything will come apart in the termination step. So, yeah, these animations are specifically prokaryotic translation, so just like with transcription and DNA replication, most information is asked about in the prokaryotic context. So eukaryotes, at the very end of 30B, the only thing you need to know about eukaryotic translation are there's like 2 slides that specifically tell you the differences. Those are the only things you'll need to know about eukaryotic translation. I'll get to that at the end. Okay, blah, 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 blah. A lot of this we've already talked about. If you want a breakdown again, here's your mRNA, and this isn't the main animation yet, but here's your mRNA. On it you have a bunch of bases. Remember that every 3 bases at a time, define something that's called a codon. And notice you go look at the anticodon, which we talked about today, is this structure here. It's a loop in here somewhere, with an amino acid on the tail end and a 3-base anticodon. So the whole point of the ribosome is to bring in anticodons to match with whatever codon it's holding on to and take away its amino acid sequence to build a protein. So, that's what's being illustrated. There are 3 spots in the ribosome. I will break down when we get to the main steps, but if you want the names right now, there's a site called an A site in the ribosome, a P site, and an E site. The A site is where you will find, to kind of give you a preview, the A site is called the acceptor site, is where new tRNAs are going to enter the ribosome. The P site is where it's going to, is the site in the ribosome where it's forming peptide bonds, because remember way back in exam 1, peptide bonds are that stuff that connects amino acids together. That happens specifically in the middle of the ribosome at a P site. And the E site, which I believe is like exit site or whatever, that's where it dumps off the tRNA once it has let go of its amino acid. So all that's preview, so now we get into the main stuff. Let's get to, okay. So, initiation. Initiation, the goal is to bring in all the pieces we need to conduct translation. So logically, we just need to think about what pieces we need. Well, obviously, we need the mRNA. That is necessary for translation to occur. And it turns out that the part of the ribosome that binds to the mRNA is specifically the small ribosomal subunit. So you need to know that in initiation, the small ribosomal subunit binds to the mRNA. We learned that for prokaryotes, it binds to a spot that has a name called the Schijn-Delgarno sequence. So again, most information is with prokaryotes. So understand that the small ribosomal subunit binds to a Schijn-Delgarno sequence on mRNA. Okay, that's weird. Binds to the Schijn-Delgarno sequence on... I hope I didn't anger FAU for downloading something, so give it a second, I guess.
You recorded it if you did.
The recording will mysteriously not be posted to YouTube if I do get in trouble. I don't think people will care, but hopefully it doesn't take too long to restart. Yes?
So was that extra stuff DTR, or is that?
The stuff ahead of, on the front end of the
Well, there's
start
like 2
codon.
sections. There's like one colored and one.
Yeah, the green one that you saw on the very left, and it also technically would include the gray spot, is the 5' untranslated region. It's part of the mRNA, but it is before the first start codon, so it doesn't really serve any purpose. The gray part is the only purpose-serving part, which is the Schijn-Delgarno sequence. So the Schijn-Delgarno sequence is before the start codon, so it won't be translated. Its job is to just serve as like a landing site for the ribosome.
But the Kozak sequence serves a different function.
Yeah, weirdly enough, in eukaryotes, the Kozak sequence is the closest thing to the Schijn-Delgarno sequence, but even I don't really understand exactly. It's not the exact same purpose as what the Schijn-Delgarno sequence serves, because in eukaryotes, the ribosome actually cares more about the 5' G cap rather than a Kozak sequence. I don't recall exactly what purpose the Kozak sequence serves in eukaryotes, but they do find that in the 5' untranslated region. Okay, so hopefully, let me see. Hopefully, it'll still have the files downloaded somewhere. Okay, read down because I don't remember the link. also really hope I don't have to re-download Adobe Flash Player. That'll be annoying. Let's see. If so, I guess I'll have to, if I do have to re-download, I guess I'll just go with the pictures. It's also not downloading, like I didn't have to manually do it, it's doing it on its own. Okay. Let me look, see if there's nice pictures here. The answer is no. I don't care, it should still work. I'm not good at drawing this, but I guess that's what we'll be left with. All right. Sorry about that.
It's okay.
All right. Oh, no, I've been forgetting to do this. No. So because of these troubles, I might also use the animation when I do the personal recording over the weekend, because this is not going to, my drawings are not going to be that good. So, initiation. We know so far we have our mRNA, and somewhere in that mRNA is a 3 base pair codon called the start codon. Okay. And ahead of the start codon, we have the Shine-Delgarno sequence. Delgarno, which the small ribosomal subunit, okay, the small ribosomal subunit initially used that as a signal to bind to the mRNA. All right. You may have seen on the animation there was a couple other things. There was a couple, I'm not going to draw them in the right spots, but there's a couple other proteins, IF1 and IF2, called initiation factors. All right, called initiation factors. So there's a class of proteins in translation called initiation factors. Their job is to help put together all these components. You will find in elongation, there will be stuff called elongation factors, and you'll find in termination, there will be stuff called release factors. You do not have to memorize the difference between initiation factor 1 versus 2 versus 3, and you don't have to memorize the differences between release factors 1, 2, and 3 in the termination step. In elongation, you do have to know the difference between the two elongation factors, which I'll get to later. Okay, so initiation factors, small ribosomal subunit, the mRNA, the Shine-Delgarno sequence. Now we need the important part, which is the very first tRNA, holding on to, in prokaryotes, something called formal methionine. It's a modified version of methionine. By formal, it's spelled like this. It's a functional group, a special functional group that is on methionine and is specifically only on the first methionine. So it's only going to be on the methionine that goes to the start codon. Like, what I mean by that is AUG can show up elsewhere in the mRNA, and regular methionine would be brought to those spots. So only the very first start codon will contain formal methionine. So it'll be a tRNA holding on to formal methionine, and of course the anticodon is going to match to the codon here. This thing is going to be brought in and told to bind there, right? So imagine that it's there. Let me see. This is so much better on an animation, as you can see, because I have to redraw everything. And the various different initiation factors will come in and leave. I don't remember which one comes in when and which ones leave, so just note that initiation factors are doing something on this picture. Again, their main job is to help put everything together. And I am missing one thing. Oh yeah, and we almost have everything. We have the small ribosomal subunit, the mRNA, and the tRNA with the very first methionine called formal methionine. The very last thing we need is the other part of the ribosome called the large ribosomal subunit. So what signifies the very end of initiation, what signifies the very last thing in initiation, is the large ribosomal subunit coming in and hydrolysis of 1 GTP. All right, so GTP is going to be hydrolyzed into GDP to jumpstart translation. So the large ribosomal subunit comes in, all of your initiation factors leave, they've done their job, so initiation factors are gone too. And GTP is hydrolyzed to jumpstart everything. Because remember, whenever you hydrolyze GTP, ATP, or whatever, you release energy. So you've now put together your ribosomal complex that's going to conduct translation. Okay. All right. Now I guess for elongation. So bottom line, both subunits of the ribosome, the very first mRNA, tRNA with the very first amino acid bound to AUG. All right, elongation. All right, so I'm going to attempt to draw it like this now. I'm making up something. I don't actually know what the codon AAA codes for, so I'm just making something up. But here's what happens. So before that got there, in elongation, the very first amino acid is actually at the P site, is at the site at which a peptide bond is going to form in a second. The A site, which is the acceptor site, is where the very next aminoacyl-Trna will enter. And it'll enter the ribosome, and we know that the anticodon on the tRNA is going to match to a codon on the mRNA, and perhaps the third base here might experience some non-canonical base pairing, as we discussed earlier. But the main goal is to bring in this amino acid. I'll talk about the elongation factors in a second, but what's going to happen in elongation, this repetitive process, what's going to happen is this methionine is going to be ripped off its tRNA and stuck onto this alanine. All right, and the animation would usually show that. This methionine on this tRNA is going to be ripped off and stuck onto this alanine, so that now you have a peptidyl bond between alanine and methionine. And then now what's left over is at the P site you have this empty tRNA that we don't care about anymore, and this slightly longer amino acid sequence on this tRNA. Yes?
So the attaching the N-formal methionine to the second amino acid, does the rRNA do that, or the protein?
The one that actually does the formation of the peptidyl bond, if that's what you're asking, is actually, and that's one of the important concepts you need to know, is actually done by the large ribosomal subunit specifically. I don't know if it's the rRNA in there that does it or the protein that does it, but it's specifically the large ribosomal subunit that does the whole forming peptide bond thing.
So it's not a ribozyme?
It might be the rRNA in there. I don't know that particular detail though. The only detail that I remember and that is asked about is it's the large ribosomal subunit. It might be the ribozyme built into it, but I don't know for sure. So this component of the ribosome is the thing that's ripping off this amino acid and sticking it onto the amino acid that's over here in the acceptor site. What's left behind again is in the peptidyl site is you have this empty tRNA, and through a process that we're going to talk about, which is a cyclic process of elongation factors, everything shifts over one spot to the left. Okay? Each cycle of elongation, bring in a tRNA, rip off an amino acid to stick onto an extending chain here, shift everything over to the left. What you end up in the exit site, or I believe it's called the exit site, is an empty tRNA that doesn't have anything on it, so it's going to get thrown away very easily. And what used to be over here in the acceptor site is now in the peptidyl site, and now you have an empty acceptor site to bring in the next aminoacyl-Trna. So elongation is those particular steps repeated over and over again. Eventually you're going to get however long of an amino acid sequence that you're supposed to have. So this process of bringing in stuff and shifting things involves proteins called elongation factors. And I always get the first two mixed up, so that's what I was hoping to use the animation for. So in fact, before, let me see if I remember. I don't remember if it's TS or TU that comes first. Let me see. It is first. TS later. Okay. TU then TS later. All right. So, how does aminoacyl-Trna even get brought to the acceptor site in the first place? So it turns out that, I guess I'll draw it down here. I guess we'll call it alanine. That every single aminoacyl-Trna is being held onto via an elongation factor called elongation factor TU. I think it stands for thermo unstable, but I don't think you have to know the acronym. But it's being held on by elongation factor TU. When it brings it into the acceptor site, GTP will be hydrolyzed to power the formation of a peptide bond. So imagine this thing's bringing it into the acceptor site. And when it brings it there, GTP will be hydrolyzed into GDP. I believe, where is that going to be drawn? I think that's actually just drawn right here. Sorry, this is what I would use the animation for. Let me make sure that is the case. Oh, maybe I can use this. Not really. Okay, 1 second. Yeah, all right, so you guys won't be able to see it, so I will just draw it out. All right. This thing, elongation factor TU, is holding onto the tRNA and is holding onto GTP. It gets brought to the acceptor site, and this GTP will be hydrolyzed into GDP to power the formation of a peptide bond. So what we have left here is an empty tRNA, elongation factor TU, and GDP. All right? Now, this elongation factor TU's next job is to eventually go find the next tRNA to guide it to the ribosome. But the problem with that is it needs to get rid of this and it needs to get rid of that. So, how does it get rid of GDP and puts on brand new GTP onto itself? That's what the animation would usually talk about, and basically the way it goes is that GDP gets replaced by another elongation factor. called elongation factor Ts. All right? That GDP gets replaced by another elongation factor called elongation factor Ts, thermostable. And almost immediately, the elongation factor Ts gets replaced by fresh GTP. All right? So there is no way to go straight from GDP to GTP. You have to go through this intermediate step where GDP is replaced by elongation factor Ts, and that one is replaced by GTP. Now that you have this, it's going to go find a nice new tRNA to bind to and bring that tRNA into the acceptor site. So there are two cycles going on in elongation. The cycle going on in the ribosome, which is bringing in this aminoacyl tRNA, ripping off that amino acid and sticking it onto this elongating chain and shifting everything over. That's one cycle that's going on. Simultaneously, the second cycle going on down here has to do with elongation factor Tu trying to replenish the GTP it is using and finding a new tRNA to bind to bring it into the ribosome. So those are two cycles going on simultaneously. And again, to summarize the elongation factor down here, elongation factor Tu initially has GTP. However, it uses the GTP to power the peptidyl formation in the ribosome. It needs to get brand new GTP, but in order to do that, it dumps GDP out, replacing it with another elongation factor called elongation factor Ts. And then fresh GTP knocks off elongation factor Ts to go into elongation factor Tu. This thing then goes and finds another amino acyl tRNA to continue the process of translation. There is one more elongation factor, and I believe, I don't actually remember the letter, it's like EFT or EFG, let me see, I never remember this one. It is EFG, okay. One more elongation factor, EFG. This one's only job is to do that shift I was talking about, that shifting, that translocation of everything moving to the left. After you've ripped off this amino acid and stuck it on here and you have that useless tRNA right here, elongation factor G's job is to shift everything over to the left. Okay, so it's involved in a process called translocation. All right? It is a different elongation factor not involved in this cycle down here. It's a completely different one whose job is to shift everything over to the left so that you can free up the acceptor site and continue elongation. I'll try to cover termination real quick because it's not that bad. But elongation are those two cycles working in tandem. I'm pretty sure you guys can use the animation. I'm sure it'll work on your guys' computers, so you'll be able to review that in more detail than what I could possibly draw. But the very last step in translation is termination. All right, so here's my attempt again at drawing. Let's see. So we have some aminoacyl tRNA. Remember that each step was attaching an amino acid to the previous, to the very next amino acid in the chain and shifting everything over. Like imagine this here represents the very last elongation step where we just shifted everything over. We're about to dump this useless tRNA and we have an empty acceptor site. The way termination works is instead of there being another codon here, instead of there being another amino acid coding codon, you will have a stop codon shows up here. One of your three possible stop codons show up here. What ends up happening in termination when a stop codon shows up at the A site is instead of a tRNA being brought in, you have what's called release factors. 1, 2, 3, whatever. You don't have to remember. You don't have to know the difference between release factors 1, 2, or 3. Just know that you have special proteins that will come in instead to the stop codon. What these release factors do specifically is they cause, and I guess I'll write it down here, they cause the large ribosomal subunit to become a hydrolase. Remember, the large ribosomal subunit's job initially was to form those peptide bonds between your amino acids. The release factors transform the large ribosomal subunit into a hydrolase. So what the large ribosomal subunit ends up doing, it cuts off this thing, and your amino acid sequence floats away. Your finished amino acid sequence, however many amino acids it is, just leaves. These release factors then cause everything to fall apart by hydrolyzing GTP into GDP, because everything needs energy to do all this. These release factors will cause the large ribosomal subunit to leave from the small ribosomal subunit. It will cause the mRNA to fall off, and as I stated earlier, this amino acid sequence is finished, and it'll also float away, and we learned in chapter 1 it'll fold into a secondary and tertiary, exam 1, it'll fold into a secondary, tertiary, and maybe a quaternary structure. All right, so termination, the way it works is instead of there being a regular codon here, it's a stop codon. Special proteins called release factors bind to this. They cause the large ribosomal subunit to become a hydrolase now, which cleaves this amino acid sequence, and it leaves, and everything falls apart, and you've ended translation. In order for translation to begin again, you must do all those things that we said in the initiation step, which is the small ribosomal subunit finding mRNA, another brand new formal methionine on tRNA, all that stuff that we talked about earlier. So we'll have to end it now. Use the animation on that PowerPoint. We'll cover this in a lot better detail than I could possibly cover. All right. I'll stick around if anyone has any questions. And